Hello and welcome to another episode of Hema Now, our haematology podcast series. I'm your host, Dr. Jonathan Sackier. And in this series, we aim to highlight the leading experts across the diverse subspecialties of haematology. Today, I'm thrilled to be joined by Dr. John Riches. Dr. Riches' research investigates the alterations in metabolism that accompany B-cell activation and how this is reflected in the metabolism of low and high-grade lymphomas. After obtaining his MD from Oxford University Medical School, Dr. Riches trained in haematology at Imperial College London. During his Cancer Research UK Clinical Research Fellowship at Barts Cancer Institute, he developed his research interest studying the defects underlying impaired T-cell immunity in chronic lymphocytic leukemia. He completed his PhD at Queen Mary University of London. And in 2016, Dr. Riches joined the Centre for Haemato-oncology in the Barts Cancer Institute, where he is now clinical reader in Cancer Immunometabolism, and he's an honorary consultant haemato-oncologist. Dr. John Riches, it's a real pleasure to have you on the EMJ Hema Now podcast. It's a pleasure to have you here. So, Dr. John Riches, a real pleasure to have you with us here today.
Great. Thank you very much, Jonathan.
Excellent. So you've had an incredible academic and clinical journey through some of the world's top institutions. What initially took you into hematology, and how did your early experiences shape your research focus? It's always nice to have an origin story.
Yes. I remember it's one of these things. I remember finding hematology actually really quite difficult as a medical student. And I decided that I would my university had these things called these special study modules. And I thought, right, I need to sort this out because I've got my university finals coming up. And I did a few weeks specifically with the hematology group at the hospital and really enjoyed it. I just really enjoyed the mix of clinical science, basic science. I love the fact that hematologists could see the patient, do the biopsy themselves, and actually look at it under the microscope themselves to see the diagnosis. And I like this really close interface between the clinical medicine and the laboratory medicine. So that really always appealed. The team there were very kind, very supportive, and all of these things combined together really sort of piqued my interest, and it was one of the things I wanted to do later on.
And what were the influences that took you down the research, the route that you went for your research?
I always, as an undergraduate, I found immunology really interesting, and I think doing clinical medicine I really enjoyed, but I really wanted to try and combine the two, simply because the you could see the potential in terms of sort of understanding the immune system, the way the immune system worked for developing sort of new therapies for patients, and I found that very motivating. So I always kind of wanted to try and combine the two if at all possible. I've just been very lucky that so far I've been given the opportunity to do this.
Excellent. So your PhD work examined impaired T-cell immunity in chronic lymphocytic leukemia, CLL. Can you talk about T-cell exhaustion and how it's relevant to clinical care? And maybe start with a little bit of context about that for those of us like me who are not that clued in.
So when I did my first job in hematology, it was on a bone marrow transplant unit, and I just thought this was the most amazing thing, that you could essentially transplant somebody's immune system, and this could cure cancer where chemotherapy has failed. And this was really before the advent of what I'd call modern immunotherapy. But I mean, this has been a major driver over the last sort of 10 years with the introduction of immune checkpoint blockade, CAR T cells, bispecific antibodies now. And of course, the flip side to this is almost by implication, if you can modulate the immune system to treat cancer, then the idea is that there's some problem with the immune system if you've got cancer. So the idea is that the normal immune system is continually eliminating cancer cells, and that if you've got cancer, that represents some sort of failure of that. And so around the time that I started my PhD, there had been a lot of work done, led by people like John Werry in the States, investigating this concept of T-cell exhaustion. I think that the name's a really good one. The idea is that it's T-cells that are chronically stimulated over many years, particularly in the context of chronic viral infections that he had studied, done a lot of work in. They start to acquire a kind of lot of sort of phenotypic and functional characteristics of being exhausted. So, for example, they upregulate lots of inhibitory receptors, they lose the ability to grow and proliferate, they can't make cytokines anymore, and they lose their kind of cytotoxic capacity. To kill off, viral infected cells. And so this is one of the reasons why chronic and viral infections become chronic. The T cells are no longer able to fight them off. And so we wondered really whether this was applied in cancer. There did seem to already be some interest that it was. And in chronic lymphocytic leukemia, we knew that there were some problems in the T cell compartment and that people's T cells didn't work quite as well. And so we just really, on the back of this work, tried to address the hypothesis that T cells in CLL may be exhausted. And so we had a kind of almost a shopping list of things we were looking out for. Did they have this? And in the majority of the situations they did have these features of T cell exhaustion. In terms of what this means for patients, unfortunately, the biggest cause of morbidity and mortality in CLL now is actually not that there's been major advances in the treatment. So the treatments now work really well, but the things that are still, unfortunately, causing real problems for patients are infections and second cancers. So the immune system's not dealing with those. They had poor responses to vaccinations, and this was really brought to the fore, obviously, over the last 5 years with the COVID pandemic. And they also have problems with their immune system going a bit skew-if and causing other things. So they have problems with autoimmune hemolytic anemias and autoimmune cytopenia. So really beginning to understand that and try and work out where the immune system has gone wrong, I think is still a really important question.
Right. So your research explores how metabolic changes drive B cell activation and lymphoma progression. Can you break down why metabolism is such a critical factor in understanding and treating lymphomas?
Well, I mean, it's just absolutely fundamental thing. For any cell that is growing and proliferating, it needs to have a metabolism that supports that. So for a cell to make another cell, it needs to be able to make a full complement of DNA, RNA, organelles, cell membranes, you name it, it needs to make it. And so it needs to have that the kind of it needs to kind of basically rewire its metabolism to sort of set that up, and a lot of oncogenes are involved in doing exactly that. And so the idea is that obviously one of the features of cancer over the last 20 years is people have done a lot of genotyping of it and a lot of genomic analysis of it, and it's just really highlighted the complexity of the problem. But a lot of these things are converging on the same sort of pathways one way or another. And the idea is that metabolism is one of these kind of key areas that a cell needs, and that if we really begin to understand it, then we can develop new therapies and look at it as developing in the clinic. And it's not just traditional pharmaceutical therapies, drugs that block particular enzymes or transporters, but also, and I think this is a really important thing that's again come to the fore over the last few years, is looking at the interaction with what people eat, people's diet. So I think it's fundamental there. It's also really important for what we do in terms of in the clinic. So we use metabolism. Glucose uptake is the basis of CT PET scanning. So you give patients basically labeled glucose. That then admits something that can be detected by these PET cameras. That allows us to see what cells in the body and where they are that are using lots of glucose, and it's a really important way of detecting and staging lymphomas and seeing basically how much lymphoma a patient has got, and also really importantly measuring response to therapy. So we talk about this term of patients being PET negative after the first few rounds of treatment, and that's a really helpful biomarker of how patients respond. So there's all of these various ways in which
Hmm. Hmm. So your work has explored how genetic changes, particularly involving MYC and P53, influence the transformation of CLL into Richter syndrome. I think I told you I'm but a simple surgeon. I had to look up what Richter syndrome was, so forgive my ignorance. Can you please first set the stage and then tell us what have been some of your most interesting findings in this area?
um, So basically what Richter's syndrome is that CLL itself is a relatively what we call a low-grade type of cancer, a low-grade type of lymphoma, leukemia. And the cells themselves are relatively small. They don't proliferate that much. It's a relatively indolent, slowly progressive thing. And it's usually quite disseminated around the body. So clinically, it tends to be quite symmetrical. If people have got enlarged glands, it's usually on both sides of the body. Richter's is where cells basically acquire another mutation that causes them, we say, to transform. They become larger, they start proliferating a lot more. And whereas CLL, you can kind of keep an eye on patients and don't necessarily need to treat them straight away. And that's a really important part of managing the situation. Richter's is something you really need to get on and treat because it will progress. And unfortunately, the same kind of genetic changes that cause the transformation often mean that it's quite resistant to traditional chemotherapies. And so I think there's a real area of unmet need there. And that idea was one of the reasons we're interested in looking at metabolism in this situation. Now, we know MYC is very interesting. And of course, it's probably the most dysregulated gene in cancer. It's implicated one way or another in up to 70% of cancers. And it does it. I mean, if a cell wants to grow and divide, it switches on MYC, basically. And MYC then controls about 15-20% of your genes and basically sets up a cell to start growing. And we know MYC is really interesting from a metabolism point of view. So one of the key things that MYC does is it switches on a cell's kind of appetite. They start taking up and eating and metabolizing glucose. They also start taking up amino acids, in particular glutamine, and they start breaking them down for two reasons. One is for fuel, but not quite so much. It seems to be more that cells need it to make stuff with. So they're using glucose in particular to make stuff with. So they're using glucose to make DNA, to make RNA. They're breaking it down. And MYC is really important in switching on all those pathways that allow a cell to do that. And so a lot of our work is focused on a particular part of that, a MYC kind of a pathway controlled by MYC, which produces an amino acid serine. And serine is just very important in terms of it supports another part of metabolism called one-carbon metabolism. And serine basically donates carbon atoms to this part of metabolism called one-carbon metabolism. And the cells then, I almost think about it sort of, if you've ever played with those like kind of Lego sets and you've got steps there where you have to add in bits one step at a time, that's kind of what one-carbon metabolism is at a time. And it's doing it one carbon atom at a time. So it's really quite phenomenal to think that cells are putting themselves together one carbon atom at a time, and MYC controls that, and that's really been the focus of our work. And we've shown that if you block various steps in that pathways in lymphoma, in particular high-grade lymphomas like Richter syndrome, then it can be beneficial, and we've seen some good efficacy. And it's something we'd like to develop further in the clinic.
Interesting. So you mentioned your interest in immunology back in the day, and with your expertise in immunotherapy and targeted treatment for lymphoid malignancies, what are some of the most promising new approaches that could revolutionize treatment in the next decade? I mean, I just want to say that the joy for me doing a podcast like this is it forces me to revisit areas of medicine that I would otherwise not revisit, and discover, my goodness, how your specialty has advanced since I graduated. I mean, the changes are phenomenal. So immunotherapy, targeted treatment for lymphoid malignancies, have at.
It's been a super exciting time, and it's really been, I mean, as the changes, even my career as a haematologist over the last sort of 15, 20 years have been amazing, and it's been great to see. And you just hope the trajectory continues because, the improvements we've seen in terms of the treatments. And it also really important in terms of how tolerable they are and the improvements we're seeing in people's quality of life. I mean, some of the treatments coming in now, barely have any side effects. They're amazing, and yet they're very effective for certain types of lymphoma. In terms of what's going to happen going forward, I mean, we're very, very much already in the kind of the immunotherapy era. And I think we will just over the next 5-10 years, it's really going to dominate a lot of the clinical trials. And a lot of the advances, I think there's no doubt about that. We're going to see, we're already seeing chimeric antigen receptor T cells come in. I think that they will continue to be developed. They will continue to get better. We will be using them hopefully earlier in people's treatment. the big advantage being that if you can get them in earlier, obviously traditional kind of chemotherapies that we've been using do have an impact upon people's normal T cells, normal immune cells, often quite negatively. And if we can get the immunotherapies in earlier, particularly if we continue to use people's own T cells, then I think that will be really helpful for people, and they will work better. The challenge at the moment, I think, is quite logistical in that you have to go through this process where you bring the patient up, you collect their T cells, you send them away, they kind of get reprogrammed, they then get reinfused back into the patients, and it's quite a bit of a to-do. And often that period can be quite a dodgy time, in that if you've got really aggressive, nasty cancer, it can progress, and you've got to think of ways of holding that, and that can be quite challenging. So I think hopefully another big advance will be looking at kind of off-the-shelf type cellular immunotherapies, where you can potentially use, perhaps other people's T cells that have somehow been switched off, so that they don't, there isn't this risk of them attacking the patient through other means. This kind of graft versus host type thing, or other types of immune cells like natural killer cells coming in. Then I think that these bispecific antibodies, sometimes you sort of wonder who came up with these things, because they're just so clever. An antibody where one bit sticks to the tumor cells, another bit sticks to a T cells and switches it on, I think is a really great idea. And we've been started to use them in the clinic where I work, and we're seeing great responses with them. They come with their new challenges, there's no doubt about that. They're available off the shelf. You can give them when you want, and I think what's also really important is that a lot of these therapies are a bit more tolerable than the old, really quite intensive chemotherapy regimens we used to have to use. So, for example, if a patient had lymphoma, we'd treated it successfully, or we'd struggled to treat it and it had come back, we're often giving people really quite intensive chemotherapy regimens with a huge amount of side effects. And that obviously limited, A, it was not very nice, it still isn't very nice, and B, it did limit the population of patients we could give them to. We just weren't able to do that in people who were perhaps that bit older, that bit frailer, which is a real struggle. But I think what a lot of these newer therapies are doing, they're a lot more tolerable, so they're expanding the area of people that we're able to effectively treat, which is great. The other area, I think in terms of targeted therapies, we've seen a great deal of success with these tyrosine kinase inhibitors, in particular targeting this molecule Bruton's tyrosine kinase, which is a key part of the B cell receptor signaling pathway, which is kind of like the on switch pathway for most types of lymphoma, in fact normal B cells as well. And we've seen great developments there. So we started off with the original one, ibrutinib, about 10, 15 years ago, and we've moved through second generation and third generation versions of this. And what's really exciting now is I think there's this new class where we're looking to degrade it rather than inhibit it, and that's a really exciting thing. That's sort of still in very early phase 1 clinical development, but the idea that you can sort of tag the cell so that it or tag a protein that the cell basically chomps it up using this part of the cell called the proteasome, the sort of the recycling center of the cell, as it were, is a really interesting idea. And it kind of works twofold. So it works firstly by blocking how that protein works, like all the inhibitors did before. But the other big advantage, or the other big thing that it has, is because it's actually degrading it and removing the protein from the cell, as if that molecule is having any other functions that support the cancer, like a kind of a scaffolding effect. And many of these molecules they kind of sit there and they act as a kind of a hub for whole pathways to kind of exist around. And if you can remove that protein, then it takes away this kind of scaffold function as well. And so the potential clinical benefit of that is enormous. And the potential for getting past, one of the features of tyrosine kinase inhibitors is that you can get mutations that block them binding and stop them from working. And I think one of the exciting things about this degrader class is that they may get around all of that as well.
I love what you said, that you wonder how people dream these things up. And I've recently been learning a lot about radionuclides therapies, tagging an isotope, a radioisotope, to a key, if you will, or or a bomber, and it's delivering a load to the specific cell and using these targeted alpha therapies. And it's like, duh, you know, I mean, I still have no idea how you actually make a radionuclide, and I still have no idea what a cyclotron actually does. But we are so privileged to work in this amazing profession where there are all these advances, and we keep making people better and better through new and different ways. So, John, you balance laboratory research with clinical practice at Barts. What are the key challenges that you face translating the discoveries in cancer immunometabolism into treatments? These days, we've got financial constraints, demands on clinicians uh, um, for workload, and then again, more financial constraints. So, how do you see this translational research?
There's no doubt about it. It's really challenging, Jonathan. It was always a difficult thing to do, and it's just getting harder. I think the multiple challenges. I mean, firstly, it's great that there's all of these advances, but there's all these advances, and you've got to keep abreast of them, and they're happening both on the clinical side and all the different types of cancers that we treat, and other non-cancerous conditions as well. There's a huge advances being made, and keeping tabs on all of this is hard, and keeping up to date. And of course, it's happening on the scientific side as well. So trying to keep abreast and just keep up with the developments in one field is hard enough, and if you're trying to do it in perhaps more than one field, it's really hard. And yeah, it's just you kind of almost have to bounce off one of the other, and just rely on your colleagues and people. You're kind of in the middle as a clinical academic, sort of learning from everybody, really, which is great fun, but also very challenging. There's no doubt that the financial side of things is challenging. We know healthcare systems across the world, of course, the NHS is absolutely no exception to that, are struggling. There's less money around from the government. At the same time, you've got an aging population, so there's more work to do, and there's less resources to do it. It's the same on the university side, of course. That's been well publicised in the media recently, with challenges with the relative slow increase in tuition fees and the problems with recruiting students from overseas and all the challenges there. And of course, that manifests itself in the universities placing more demands on their academics to teach and perhaps giving them a bit less time to do research. So there's all of these challenges there. There's also, I mean, the other big area is in terms of funding for research itself. So, I mean, the funders, the charities, the research councils that pay for the researcher, they're also experiencing financial problems. There was issues after the COVID pandemic. The charities couldn't raise as much money, clearly. And so, there's no doubt about it that the research grants that we get aren't really keeping up to speed with the rises in cost of living, and we're seeing increased costs of paying people's salaries, of buying research reagents, of doing the experiments. So whereas the grants aren't increasing as quickly, and so we're having to try and do more with less in terms of research as well. And in terms of actually publishing, it's great, but the range of scientific techniques and amount of data that you can generate, and the amount of data that you find in a scientific publication now it feels like it's almost doubled over the last sort of 10, 15 years ago. In fact, my PhD supervisor, always sort of says you tend to find more data in the supplement now than you did in the original paper from 10, 15 years ago. And it's just amazing how much stuff you have to put in for the same sort of level of publication, although undoubtedly the quality of the science continues to drive things forward. So, yeah, there's all of these struggles. I think within metabolism in particular, there's lots of challenges there. I mean, I think the thing that there's the two main challenges, I think, are keeping it specific and trying to keep the cancer cells, because clearly every cell in the body is metabolizing stuff, so you need to try and look for that therapeutic window. And then the other thing is that kind of metabolic flexibility, and cancer cells are no different. And you can very effectively block one pathway, and they just go, right, I'm just going to start eating something else, or switch on something else, or switch on another enzyme or an isoform of the same thing. So, there's there's challenges across the board.
I mean, one of the financial challenges that I think um, is creating a field change is the cost of university overheads to grantors. I mean, practiced half my career in the United States, and 60% overheads were typical, and then I learned that they're nearly double that here. And what we were seeing in America was a lot of research. Researchers were moving out of the university environment and setting up either with private labs or setting up within a company and discovering that they could do it at a fraction of the overhead. Um, there are competing interests from the university as to what the money is used for. And yeah, we've got to work out what our universities exist for. Are they teaching institutions? Are they clinical institutions? Are they research institutions? Because I don't think they can be all three in this day and age. So challenging. So you mentioned, going back to the science, that studying B cell metabolism could have implications beyond cancer, possibly autoimmune diseases. Can you share some insights into how this research could extend into other fields?
Yeah, I mean, I think one of the things about sort of, let's say, one of the challenges I was talking about was this therapeutic window. So we always wanted to study kind of what was going on in normal B cells, in the normal lymph node, in the normal part of the lymph node, the germinal center, which is the sort of one of the major areas for the it's got a kind of your humoral antibody immune response. And of course, we do see, obviously, a lot of the lymphomas do arise from these B cells at this particular stage. And there's, all sorts of interesting sort of reasons for why that might be, including the kind of the natural genetic instability that's a key feature, an evolutionarily driven feature of the normal immune response. And so studying normal B cell metabolism, in particular germinal center metabolism, has always been a key aim. And we've found that there is this impact. If you do target these metabolic pathways, you can affect normal immune responses, the generation of normal antibodies to antigens. And so automatically you think, well, these could be used for autoimmune diseases. When I speak with colleagues that treat either neurologists or rheumatologists that are treating things like MS or rheumatoid arthritis, they are using, in many cases, much of the same armamentarium in terms of treatments as we do as lymphoma doctors. Our primary aim is to calm down and switch off and kill aberrantly activated lymphoid cells, lymphocytes. And so they're using things like steroids, methotrexate, rituximab. So a lot of the developments that are happening in terms of lymphoma can be useful for treating patients with autoimmune disease, and almost certainly the other way around as well. I don't think metabolism is any different. If we find a drug that can treat lymphoma, then you would hope that it might be also useful for say treating rheumatoid arthritis. And that's quite a good example, because a key drug that we're very interested in terms of our research is a drug, methotrexate, that works by targeting folate metabolism. And of course, that's used very widely for autoimmune diseases, in particular rheumatoid arthritis. So there's hopefully a great synergy there. And yes, we try to work closely with our rheumatology colleagues to develop that as well.
So watch this space. Switching subjects, I saw that you won a Barts charity small project grant investigating the potential of breath biopsy in hematologic malignancies. And this fascinates me. I've been involved in looking at a couple of other projects where people were using breath biopsy for a range of different things, less invasive means of diagnosing disease earlier. Talk us through what you found.
Yes, this has been sort of very interested to get involved with, it's been quite challenging, but good, but yeah, very sort of rewarding. I had a colleague who I was working with in a previous lab who was going to join this company, who were developing breath tests, and so we thought I sort of had a look at the time. I thought, this is interesting. I don't think anybody's done this before in lymphoma, and so we thought we would do it, and we did, and they were very helpful and collaborative. And we collected some breath samples. We tried to start the study. We had planned to start the study in February 2020. And I can tell you that collecting people's breath samples in the COVID pandemic was a bit of a non-starter. So it took us quite a lot longer than we would have liked. But we did do it. And we did find that in the breath of patients with lymphoma, they have increased levels of these things, methylated alkanes. And what was quite striking about that, in things like sort of heptane, hexane, decane, things like that, you seem to more think about them in the context of sort of petrol products than you do in cancer. But what was very striking was that another group in the States had really sort of pioneered this area, had published this paper in the Lancet 25 years before using a completely different technology in lung cancer. And they'd found pretty much exactly the same chemicals at increased levels in the breath of patients with lung cancer. This has been taken forward, and I think we're getting very close now to having a kind of a clinically useful test now for lung cancer. And we're kind of hoping that we might be able to piggyback off the back of that in lymphoma. And the aim really would be, I mean, we're at a very early stage in terms of lymphoma. We've just looked at it in a pilot study. There's a long, long way to go. But what I hope we can potentially do is have a quick kind of almost like a breathalyzer that you could have on your desk in clinic. You can have a patient, you could ask them to take a 5-10 second, completely non-invasive breath test, exactly the same sort of idea as, a policeman breathalyzing somebody for alcohol by the roadside. Very quick test, completely non-invasive, that could give you a readout of, whether they had lymphoma or not, with the idea that, these are volatile organic compounds we're talking about here. So wherever they're produced in the body, they'll go into the bloodstream, and then you will excrete them in your breath. You can excrete them, of course, in sweat and urine and anywhere else as well, but they will be in your breath. And if you can pick those up, then you could use that to potentially either pick up lymphoma or to monitor people that have had it to see whether it's gone away or whether it's come back. So there's all sorts of potential clinical applications. And I think it'd be a nice counterpoint to those more laboratory-based techniques that can take sort of hours, weeks, days to come back. A lot of them that come back, essentially, you can actually give people some information very quickly, and also non-invasively without doing anything, sticking any needles in them or anything like that. So I think there's great potential there, but we're a long way away from the clinical application. There's always going to be all of these questions about sensitivity and specificity, and we've really got to address those. Yeah.
Sure. I was talking to someone who recently about dogs and the diseases dogs can detect. And there seems to be validity to this, that they actually can detect people with various malignancies. And I wonder if we know that dogs are capable of smelling in far more minute quantities than we are, and also sort of create timelines of what they can smell. So, we know about CAT scans. We could end up with dogs.
Cats, yeah, absolutely, yes.
It's a fascinating space. I mean, the idea that we could be just checking someone's breath as part of your annual physical, and if you're detecting abnormal things, you've got a basis for further investigation. Well, again, watch this space, or maybe smell this space. So I would like to end, John, with a fanciful question. If you had 3 wishes for your area of healthcare, what might they be?
I think the first one would definitely require a genie. It's we've got to look at people's diet, and do something about that. And there's been a lot of interest over the last couple of years in ultra processed foods, high sugar, and I think that from sort of studying cancer metabolism, immunometabolism, I think it's pretty clear that what we eat is fueling the cancer cells, probably fueling the development of cancer. And then actually, if we could get people on healthier diets, maybe particularly tailored diets, depending on exactly what cancer they've got, what the genetic changes are driving them, we may be able to have a therapeutic impact on cancer. And I think that would be a really exciting thing to look at. So yeah, we've really got to address that. And the challenge is it's so ingrained. I mean, if you kind of wanted a kind of a rundown of the foods that we shouldn't eat, you could probably just pop down your local newsagent or corner shop and you'll find it chock-a-block with sweets and chocolate and crisps and cookies and probably cigarettes and alcohol as well. And that it's the whole food industry is kind of set up to give us what we want rather than what our body needs. And I think encouraging people to eat less processed foods, eat more fresh fruit and vegetables, perhaps reduce their meats, try and cook more at home. There's huge challenges there, and it's going to be a combination of education. I think simple messages. I like the one. I think it was Tim Spector maybe, was talking about sort of eating the rainbow. If your foods are naturally brightly coloured, they're brightly coloured because they evolved to attract animals to eat them. They're probably good for you, as long as it's natural colourings, not artificial colourings. And you could counterpoint that with bin the beige, I think. So a lot of bad sort of foods are beige coloured. So if you're eating lots of pizza dough or burger buns or crisps or noodles or whatever, then they're all roughly the same colour. So simple messages to sort of educate people, I think, could be really effective and improve people's health. Not just in terms of cancer, but general health, cardiovascular health, obesity, diabetes as well. And then I think governments have got a role to play in terms of regulating the food industry, taxation, removing taxes from good foods and increasing the taxes on the ones that are less good for you. So I think that would be genie wish number one. I think you probably do need a bit of magic for that one. Number two, in terms of studying metabolism, what's really come to the fore over the last 5-10 years is the importance of studying it in the physiological context. So traditionally, a lot of work's been done in these, where you grow cells in a test tube. Grow these cancer cell lines in a test tube in a specialized medium, and this medium is really kind of like a nutrient-rich broth that gives the cancer cells everything they want and bears very little resemblance to what is actually going on in the body. And there's been some major advances with people started to look at the metabolic behavior, I suppose, the metabolism of cancer cells in media that's more like people's plasma, so blood plasma, and all sorts of things have come out of that already. But there's so much to understand more about. So looking at other spaces in the body, what the normal concentrations, in your lymphatic fluid and the fluids in your various organs, how does that change? Fluid in the brain, and understanding how cancer cells adapt there. There's a long way to go. So to understand more about metabolism in normal and how it relates to normal physiology and how we can modulate that physiology with diet as well as a treatment, I think would be super cool. And I think really important. And the last one, I think, it'd be nice. I think when I came into kind of the immunotherapy sort of side of things, I was looking at kind of targeting the immune system in cancer and understanding more about the immune system in cancer. We're kind of this was sort of 2007 time. There was great promise, but it hadn't yet delivered really very well. We had bone marrow transplants that we knew, and we had sort of immunomodulatory drugs like thalidomide and lenalidomide in the blood cancer space. But there was a lot of interest. There've been a lot of advances in the basic science, but we hadn't quite got there yet in terms of clinical. And that's probably roughly where we are in terms of metabolism right now. The whole landscape's changed over the last 15, 20 years. And there's loads of work going on now, and it's all very exciting. But we just haven't. There's been a few clinical successes, but it's been pretty limited. And so what I would really hope is the genie could magic up a few very effective clinical trials of this stuff, because I think that would obviously be great for patients. I think there's a huge potential there, particularly in prevention as well as treatment, looking at diet and people's lifestyles as well and how that relates to the risk of certain cancers. But obviously it'd be nice to see the success, all this research be translated into the clinic.
Yeah, absolutely. Well, three very interesting wishes. I will say at one institution I worked, no name, no pack drill, as they say, the dean, who was a big believer that the impact of diet on disease, endeavored to change what was sold in the vending machine in the cafeteria, the staff cafeteria. And there was a
Yeah.
revolt. He tried to get rid of, I love that expression, bin
Right.
the beige. He tried to get rid of that stuff and introduce more healthy foods. And basically people said, we'll go on strike. If you do away with our burgers and chips and pizzas, we will go on strike. So sad state of affairs. I'm afraid that's all we've got time for today, but I'd like to thank Dr. John Riches. Thank you for being with us, for all the work you're doing, both in the laboratory and on the wards in looking after patients with these wretched diseases, and for the advances you're making to treat them. Thank you
very much. It's been an absolute pleasure. Yeah, really nice speaking to you. Thank you.
Well, I hope you enjoyed that episode of Hema Now, our bi-weekly podcast on haematology. Please consider subscribing on either Apple Podcasts or Spotify, or frankly, wherever you get your podcasts, so that you never miss an episode. Like us on social media, tell your friends and colleagues, and please check out the archives because they're growing if you've missed any episodes. And please join us again in 2 weeks for another episode. Until then, I'm Dr. Jonathan Sackier. I'm proud to be your host. Please, everyone, stay safe, stay well, stay curious. Bye for now.